I promised I'd teach you folks how to use Excel. We didn't have time in class, so I'm going to do a quick screencast here now. Excel is a spreadsheet software. It uses cells instead of Microsoft Words, for example, page layout, and in these cells you can enter numbers and text. You can perform math on those cells, and you can also input data in order to create visualizations like graphs. I'm going to teach you exactly what you need to know for this lab. So for lab number 7, enzymes, you need to make one graph, which is going to be the elapsed time to the negative for the Benedict's test as a function of enzyme concentration. So it's on Y and X respectively. So what we need to do is, this is your X-axis here, the data in X, and then the data on your Y-axis. So right here, I'm going to do the enzyme concentration, and the units are percent volume on volume. And then for the second column B, we're going to do elapsed time to change, and then the units here are seconds. You can see this gets kind of cut off. You can double click the boundary between the columns in order to have it snap there. The data that I'm using is what I got when I took this lab. If you use this data, it's an academic integrity issue. Please use your own data. Do not use mine. So we had 4 different concentrations where we're expecting results. Enzyme concentration 10%, 5%, 2%, and 1%. Then the times that I got are 35 seconds, 135 seconds, 360 seconds, and finally 720 seconds. So it's easy enough to input that data like that. We then are going to select it all by dragging from the top left to the bottom right, clicking Insert. Excel has this recommended charts function. That's not what we want today. In general, for a lot of the graphs you'll be doing, you're going to want an XY scatter, and I also want the lines connected, but I want them with smooth lines, but I want those markers as well, which is the second option here. So you'll see that Excel is going to make you this very nice graph. It's good. It's a good start. It doesn't have exactly what we need. So in Excel, you can click on what they call chart elements, just different parts of a chart, to edit them or delete them. I don't want a chart title, so I can click it and hit the delete button. That's different from other classes. For other labs, you will want a title. Here, because you're going to have the graph 1 or figure 1, and then a small, very short description as described in the lab manual, we don't actually need a title on there. We also don't want grid lines. The purpose of these grid lines would really be to help people actually pick off individual data points, like their actual values. We don't want that here. For most graphs in science, you're not using them to pick off individual data points, you're using it to visualize a trend. So I'm going to click these grid lines here, just one of them. It'll select all of them. You can see with the blue dots at the top. Hit delete to get rid of them. And then once again, the horizontal ones, click delete, get rid of it. We're missing some stuff though. We're going to click add chart element. We do want axes titles. We want the primary horizontal, primary vertical. And we can really just copy and paste these guys in here, which we wrote earlier: enzyme concentration, percent volume on volume, and elapsed time to change. Just going to hit Control or Command A to select it all, paste it. So we have now our axis titles and our units, very importantly. What we're missing, or we're not really missing anything else. There's a couple stylistic things I want to teach you. You can click on one of these trend lines tiers to select a certain data series. What I want to do is, for the, that's just the line specifically, you can change the dash type. So I'm going to use this type of dash, and I'm also going to change the color to black. And then for the markers, so I'm going to click the actual little marker right there. I can click marker up here. I can change the, this is the border, so the outside of it. I can change that to black. I can also change the fill to black once again. And I can even change the way it looks. So I can click built in. And then if I want, let's say, a rotated square, I forget what it's called, whatever. So with that, the reason I want to do those changes is that when you make graphs that are a little more complex, that have multiple data series at once, so for example, let's say you had like a couple different elapsed times for, I don't know, like you did something to the enzyme, That way you would be able to distinguish them if you printed it out on black and white using a black and white printer. That's what most of the time what scientists will do when they're reading a paper. So it's really important that all of your graphs are interpretable if you print them out in black and white. You can do so by just changing the marker style and then the line style as well. Final thing, I really dislike how these axes are, they're gray instead of black, so I'm going to change it to black right here, and I'm going to make that 1 point, same with the horizontal one, black and width up to 1 point. So you can see they are a little more pronounced now. You can see the data very clearly, and then from there you can right-click and copy that into your Word document. You can adjust the size there, and then you're good to go.